So the catabolism or the breakdown of TG is under the control of two types of lipases. That is the hormone sensitive lipase and lipoprotein lipase. So the function of these two lipases are entirely different. The lipoprotein lipase is responsible for breaking the lipoproteins and releases the contents out of the lipoproteins. So the lipoproteins are a class of molecules which are responsible for transporting the lipids from one side to another as they are insoluble in aqueous medium. So there are different types of lipoproteins starting from chylomicrons and ending up with the HDL, and free fatty acids are also mentioned whenever we say about the lipoproteins, even though it's not a pure lipoprotein, okay, because they are not having any protein part. But they again the free fatty acids are transported with the help of albumin also, albumin, the protein albumin, but they are not, the free fatty acids will not be included in the lipoproteins. So the lipoproteins, in fact, they are actually chylomicrons, the VLDL, the IDL, the LDL, and the HDL. Okay, and the chylomicrons are responsible for taking the dietary lipids from the intestine after their digestion and absorption and the resynthesis of TG. The TG is taken, TG along with cholesterol, phospholipids, right? So all these molecules are taken from the intestine to the liver for further utilization. It's carried out only by chylomicrons. At the same time, the endogenous TG, that is the TG which are synthesized in the body, not derived from the exogenous sources, like we have already stated, that is the lipid synthesis or the TG synthesis can happen in almost all the tissues of the body, especially liver, intestine, and adipose tissue. Right, so there also the TG is being synthesized because dietary lipids cannot meet all the demands of the body. So the body itself can synthesize the triacylglycerol with the help of glycerol 3 glycerol kinase enzyme, and also by the addition of different types of fatty acids onto the glycerol skeleton, right? So these endogenous TG thus synthesized by all these enzymes will be taken from the extrahepatic tissues to the liver or from the liver to the other tissue, extrahepatic tissues. All these are carried out by the other types of VLDL. So the VLDL, IDL, HDL, etc., LDL, etc. Right. So what is the function of VLDL? Very low density lipoprotein. The very low density lipoprotein transports the TG, the endogenous TG, from all the remaining tissues or the extrahepatic tissues to the liver, right? So from the extrahepatic tissues, it is taken into the liver with the help of VLDL. So VLDL transports the endogenous lipids while chylomicrons transports the exogenous TG lipids, especially the TG. Okay, then what is the function of LDL and HDL that we come to explain when we are speaking on the lipoproteins. Lipoproteins itself will be taken as a separate class. So I was just mentioning about all these lipoproteins because we have come across the enzyme lipoprotein lipase, right? So there are two types of lipases, hormone sensitive lipase and lipoprotein lipase. And it is a hormone sensitive lipase which is responsible for regulating the synthesis and breakdown of what? The triacylglycerol. So hormone sensitive lipase is a covalently modified enzyme. It can undergo phosphorylation and dephosphorylation. So the hormone sensitive lipase regulates the metabolism of TG. Right? How? Because the enzyme can exist in two conformations, the phosphorylated form and the dephosphorylated form. And the phosphorylated form of the enzyme is the active form of the enzyme. Okay, so the phosphorylated form is the inactive form and the phosphorylated hormone sensitive lipase is the active form. So how is it shuttling between this active form and the inactive form? With the help of hormones. So it's the hormones that control the action of the enzyme. That's why it is known as, the lipase is known as hormone sensitive lipases. Now, which are the hormones which are controlling the lipase action? It's nothing but the insulin and the anti-insulin hormones, right? So the insulin always, what does the insulin do? Insulin inhibits the lipolysis while glucagon favors the lipolysis. Then we'll come to know that how is it shuttling between the phosphorylation and dephosphorylated form. We know, we have already studied the mechanism in other metabolic pathways. So there is an enzyme known as protein kinase A. So what does it do? It is kinase. So what does it do? It phosphorylates, right? So the inactive or the dephosphorylation form of hormone sensitive lipase whenever the TG breakdown is required. So when there is a energy need, when there is a depletion of glucose in the body or glycogen store is depleted, what happens? The hormone sensitive lipase will be shifted from its inactive form to the active form because there's an emergency for energy. So immediately what happens? The TG in the adipose tissue start undergoing breakdown. So how it is getting broken up? It is broken up with the help of hormone sensitive lipase. So the hormone will be shifting the inactive or the dephosphorylated form to the phosphorylated form. And this dephosphorylated form to the phosphorylated form is happening with the help of the enzyme protein kinase A. Right? So the cyclic AMP comes, the protein kinase A converts ATP to ADP. So this is a inactive form which is converted ADP to ATP to ADP. So what happens? The hormone from the inactive form accepts a phosphate molecule from the ATP. So that hormone is made active and ATP is converted into ADP. And what happens whenever the energy demand is to be terminated? If the demand is stopped, what is to be done? The lipase from the active form is converted into the inactive form by the enzyme protein phosphatase. So this shuttling between the inactive form to the active form and from the active to the inactive is under the strict control of hormones. So epinephrine, glucagon, etc. enhances the lipolysis while insulin always decreases lipolysis. Okay, because insulin is it's a hypoglycemic hormone. Like glucose level കുറയ്ക്കുക എന്നുള്ളതാണ്. അത് ആവശ്യത്തിന് ബോഡിയിൽ ഉണ്ടെന്നുണ്ടെങ്കിൽ ഫർദർ അത് വേണം സ്റ്റോപ്പ് ചെയ്യണം. അപ്പം അതുപോലെ ലിപ്പോലൈസിസ് എന്തിനാണ്? whenever there is an energy demand ഉണ്ടെങ്കിൽ മാത്രമേ what TG has to be broken up, right? Otherwise it will be stored as such, right? So insulin always blocks the hormone sensitive lipase while glucagon, that is anti-insulin hormones, favors the lipolytic action by increasing the cyclic AMP concentration. Cyclic AMP level ATP will be converted into ADP and the enzyme becomes active. So it is catalyzed in the plasmale. Plasmale is the tissues hormone sensitive lipase action. If the lipolysis has to take place in blood, it is carried out by lipoprotein lipase because blood the lipids are in the form of lipoproteins. So we have to first break up the lipoprotein. Protein lipids encapsulate lipids because lipids are insoluble. So the insoluble form of the lipid is surrounded or it is enveloped by the soluble proteins. So the protein layer has to be first broken up. And that breakage is carried out by lipoprotein lipase. So that's the basic difference between the two types of lipases. The hormone sensitive lipase carries out the lipolysis in the tissues, while the lipoprotein lipase carries out the lipolysis in the plasma. So substrates are chylomicrons and VLDL because dietary TG is carried out by chylomicrons, while the endogenous TG is carried out by or transported by VLDL. So these are these two are rich in TGs. Of course, TG matra illa chylomicrons cholesterol is there, phospholipids is there, but the majority space is occupied by TG. Because TG ne transport cheyanulladana chylomicrons inde VLDL inde function. But dietary TG ne chylomicron matra kondollu, ade samayam endogenous TG ne VLDL matra transport cheyu. So lipoprotein lipase is again inactive form la irikkum. Whenever there is a necessity, avade it is lipoprotein lipase control unit is by heparin. So the endothelial blood vessels lipoprotein lipase it is attached to the surface of endothelial surface of the capillaries, and these lipoprotein lipases will be released by heparin. This heparin is converting the inactive form of lipoproteins to active lipoprotein lipase by apolipoprotein C2 and phospholipids. So they are present again upon lipoprotein C2. When we come to the individual lipoproteins, you come across this terminologies and all. Apolipoproteins means it is an apoprotein. So what is an apoprotein? That I'll discuss in the lipoprotein. Okay, lipoproteins are detail that I'll teach you. So you just try to understand that the lipoprotein lipase is in the inactive form. Normally, blood transport chylomicrons VLDL normally disturb if there is any emergency arising, then what happens? Heparin comes and causes the activation of lipoprotein lipase. How? By activating apoprotein. It's an apoprotein C2 and phospholipids. Right? When the chylomicrons and VLDL reaches the tissues, the lipoprotein lipase is activated by ApoC2 and phospholipids, and they are hydrolyzed by the active enzyme, and free fatty acids are released, which are taken up by the tissues, while glycerol goes to the liver. So in the liver, what happens again? Glycerol will be utilized. Glycerol will be converted into glycerol 3 phosphate. This all we have discussed. That is, so that I hope we have understood. So this is how the lipoprotein lipase and hormone sensitive lipase functions. Hormone sensitive lipase acts in the tissues, carries out the breakdown of lipids by, again, under the influence of the hormones. There are two categories of hormones: insulin and anti-insulin. Insulin always inhibits hormone sensitive lipase. That is, always keeps the hormone sensitive lipase in the inactive form, or it always keeps the hormone sensitive lipase in the dephosphorylated form, right? And once the emergency arises, this inhibition is removed by glucagon and epinephrine, right? Or glucocorticoids, etc. So that is, anti-insulin hormones comes into action, and what happens? They carries out the activation of the hormone sensitive lipase by increasing the levels of cyclic AMP. When cyclic AMP level is increased, ATP will be converted into ADP, and the enzyme is changed or converted from the inactive form to the active form, right? That is how the hormone sensitive lipase function. While lipoprotein lipase are present on the walls of the blood vessels. So on the blood vessels, whenever there again, if there is an emergency, what happens? These lipoprotein lipase will be acted upon by heparin. So heparin causes the activation of lipoprotein lipase, and ApoC2. It causes the activation of ApoC2 and phospholipids, and carries out the release of the contents within the chylomicrons and VLDL. That is, contents. What is the contents within the chylomicrons and VLDL? It is a TG. So TG will be released, and the TG will be acted upon by the enzymes, lipases, and causes the degradation. This is a minor path, that is the 2-MAG pathway happening in the intestine. That is, intestine, TG in the intestine. We are having the monoacylglycerol. So monoacylglycerol will be acted upon by lipase, synthase enzyme, fatty acid transferase, to give DAG. DAG will be finally converted into TG. Okay, this is only the pictorial representation. Monoacylglycerol or monoglyceride, free fatty acids. The intestinal lumen absorption. Then monoacylglycerol is converted to TG by the addition of fatty acyl CoA in a stepwise manner to give TAG, and this triacylglycerol will be packed into the lipoprotein. What is that? The chylomicrons, because it is an intestine. So intestine contains exogenous TG, right? So chylomicrons pack and they are released into the circulation, blood അല്ലെങ്കിൽ lymph. Because blood lipids ആണ്, so they are bloodinekka kudal avaru lymph ilana transportation will be taking place. Now what is happening during the fasting condition? What is the adipose tissue metabolism, or what is the fate of adipose tissue? TG, depot fat in the adipose tissue, when a person is undergoing fasting. So fasting la the glucose illa, right? So the TG synthesis എന്ത് ചെയ്യണം? Synthesisന്റെ rate അവിടെ കുറച്ചിട്ട് breakup in the proportion അങ്ങ് കൂട്ടും, right? So of course synthesis will be happening there. Synthesis will be happening only in a side by side manner. പക്ഷെ സിന്തസിസിനൊക്കെ കൂടുതൽ എന്താണ് അവിടെ നടക്കേണ്ടത്? ലിപ്പോലൈസിസ് ആണ് നടക്കേണ്ടത്. So that there is more and more free fatty acid synthesis happening, right? So the free fatty acids then will be entering into the beta oxidation. Acetyl CoA will be produced. This acetyl CoA enters into a TCA cycle. TCA cycle will be converted into or entering into electron transport chain. This is how the energy is produced. Clear? That is, under conditions of fasting. Fasting എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ എന്താ? ഭക്ഷണമില്ല എന്ന് വെച്ച് കഴിഞ്ഞാൽ ഗ്ലൂക്കോസ് ഇല്ല. അപ്പൊ ഗ്ലൂക്കോസ് ഇല്ലെങ്കിൽ എന്തുണ്ടാവില്ല? എനർജി will not be there. So immediately body എന്ത് ചെയ്യാൻ തുടങ്ങും? Next source എന്താണ്? It is a depot fat. Adipose tissue ഉള്ള ഫാറ്റിനെ ഡിപെൻഡ് ചെയ്യും for energy production. How is the fat converted? The TG start undergoing breaking up. Catabolism starting. So TG ആദ്യം DAG മാറും, DAG MAG മാറും, MAG will be converted into free fatty acids. These free fatty acids will be converted into